So let's kick it off now. Thank you so much. So I want to get Prantik here, Kundu. So Prantik will be presenting our keynote speaker. Prantik is a junior faculty member in working with us. He has done great work in the aspect of analyzing complex fMRI data and trying to get quantitative imaging in the brain and in neuropsychiatric diseases. So Prantik. Yeah.
Good morning. It is a great pleasure to introduce our keynote speaker, Dr. Bruce Fischl, from the MGH Athenula Martinez Center. Dr. Fischl is currently a professor in radiology at Harvard Medical School, a neuroscientist at Mass Gen, and he's the director of the Computational Core at the Martinez Center. Dr. Fischl's work is based on his key contributions that he's most known for, and the contributions that have been very critical, is in construction and utilization of geometrically accurate and topologically correct models of the human cerebral cortex. Many of you might know this and have used his tools in FreeSurfer, and it's a vital tool for a wide range of neuroimaging analyses, and it's a key technology in the Human Connectome project, and fundamentally it allows a anatomical image to be analyzed for morphometry to build a cortical surface model, which then can be used in many different ways, which Dr. Fischl, I'm sure, will elaborate on. In general, the work has been extremely influential, and essentially everyone uses these tools. With that, I'd like to introduce Dr. Fischl.
Good morning. So I wanted to start with my standard caveat that my natural metabolic pace is exceedingly fast. I will fly along. If there are questions that you have, please interrupt me, raise your hand and ask questions, and I'm happy to try and slow down and answer them. So I think in general, I have no slides up. So in general, I kind of have two major points that I wanted to make today. And they're really kind of two sides of the same coin. Kind of bit good news, bad news, if you will. If you take a step back and you think about what we can do with medical imaging, it's really astonishing that we can generate images of the inside of the body, the inside of the head, relatively undistorted three-dimensional images that show us the interior of an object. And that's great, but we as human beings are terrible at visually inspecting serial sections and reconstructing 3D structure in our head. There's no reason evolutionarily or phenomenologically why we would be any good at this. There are no everyday tasks that require us to look at 2D slices of something and then think, oh, what would it look like if I could assemble it back in 3D? Right? So we're awful at this. Anyone who has ever tried to manually label anything in a medical image knows that this is the case. Right? We do it in the coronal view, and then we flip it sagittally, and we're like, wow, it looked great coronally. Now it looks awful. And there are a subset of us who are less terrible. We call them radiologists. And radiologists are great at this task relative to baseline, relative to, me. But in an absolute sense, none of us are any good. And this is a missed opportunity translationally, I think. Even the best radiologists struggle to see changes in images that are on the order of 20, 25%, right? There are no clinical protocols for what to do if a tumor volume changes by 7%, because we can't see 7% changes, we can't see 12% changes, we can't see 15% changes. The change gets to 20, 25%, now all of a sudden, maybe we want to change chemo or something. But computationally, we kill ourselves to see changes on the order of 1%, 1.5%, 2%. And so there's this kind of disconnect between our capabilities from a computational standpoint and what's standard practice in the clinic. And this is a shame. I think that we on the computational side have not provided an infrastructure for clinicians to make decisions that could radically improve their ability to make accurate diagnosis and staging. So that's kind of the downside, I guess. The upside is that the anatomy matters. And having models of the anatomy allows you to do analyses that would otherwise be completely inaccessible to you. And so even if you don't care about anatomy, even if anatomy is just the wallpaper that you want to project your pet image on, having access to the models of the anatomy allows you to get far more out of your data. And I'll try and make this case. I'll show you this in a little bit. So here's an outline of my talk. I have 5 different sections. You'll note an additional word here from what I actually presented online. So first I'm going to talk about subject motion. Subject motion is ubiquitous and pernicious in medical imaging. I am absolutely certain that I have published results that are totally incorrect and only due to subject motion, 10, 15 years ago when we didn't realize what a problem this was. If you're doing medical imaging today, you must deal with motion. And this is a particularly awful confound because it's almost always the case that motion is correlated with what we're interested in, right? Little babies move more than older kids, and people who are going to get Alzheimer's move more than controls, and elderly move more than young. And so we're left in this position where the thing that is corrupting our image is correlated with the thing that we want to study. And so we've worked a lot on trying to remove motion both within and across session. Then I'll talk just a little bit about constructing anatomical models from MRI. This stuff is kind of old, but it's the basis for a lot of things we do. I'll discuss how we can use these anatomical models to guide molecular and functional analyses. Then I'll just briefly discuss a little bit what we're doing on the diffusion front and how we generate models of major fiber tracts in the brain. And then finally, this is kind of my most recent research, is what we do with architectonics, and I'll define what this is if people don't know. Okay, so within and across subject motion. So probably many people in this room have collected data that looks like this. And this is what happens if you collect an MRI scan and your subject moves around while you're acquiring the data. But in MR, we are almost always sacrificing signal for contrast. We're almost always waiting around for magnetization to recover or decay or do something that enhances the contrast of our images. And if you're a subject in an MRI experiment, I encourage you to sit and listen for when there's dead time. Is the scanner going constantly, or can you hear kind of moments of silence between the clicks? And if there are moments of silence, that means that there's an inefficiency in the sequence that you're being scanned with. It means that something could have been collected during that time. And for us, the something that we collect is a low-resolution image of the head that allows us to track what the head is doing within the coordinate system of the scanner. And this allows us to take this type of data and get a relatively clean scan. So we embed these low-resolution images that enable us to compute the 6 degrees of freedom, translations and rotations about what the head is doing within the coordinate system of the scanner, and then we update the gradients to account for the fact that the head is moved. And this results in a pretty nice image. You may not be able to see it, but this one is still somewhat motion corrupted. There's still some ringing in this image. And I should also point out that this is really mostly Dylan's work with Andre, who's in my lab. And the problem with motion is that if the motion occurs while you're reading the signal, it's very difficult, if not impossible, to correct. Right? The thing that you're reading that is going to allow you to say where the head is in the scanner is itself corrupted. So how can we deal with this? Well, the solution that we've come up with is to detect the corruption by seeing where the head was before we started to read and after we started to read. And this allows us to score every little bit that we acquired and decide whether anything in the intervening spaces were corrupted. And so here's what we do. We collect lines of data as usual in MR. Then we can score them. We can say, well, for this line, I can see the head was here at this point and at a very different point there. So I can infer that motion occurred while I was reading this little bit of data. And then on the console, you can just say, I want to replace the worst 10%, or you can set a threshold with the amount of motion. And at the end of the scan, we'll go off, get rid of these, and reacquire them. And much of MR these days is done in 3D. We're actually acquiring data from the entire image volume. This is by far the most efficient way to acquire data. And the downside of the 3D acquisitions is that even little bits of motion anytime, anywhere during the scan can corrupt the entire scan. And so that's what's shown here. So I'm going to show you a little movie. Can we turn the lights off at the front, or is that not possible? So here's a motion corrupted scan, and I'm just going to show you a movie in which I iteratively take out one line and replace it with another one as I go through the sequence. And so you can see we can kind of iteratively clean up the scan until with about a minute, a little more than a minute of reacquisition, we get an absolutely crystal clean scan. Okay, any questions on that stuff? So a second question is what we do across session. We would very much like to have our slices be, for example, in the same place across session so that we can maximize our ability to detect changes from one scan to another. There are a couple of problems with this. Even if we try and align across time, the alignment itself is not really a rigid one. A rigid alignment doesn't describe, that's all right, you can just leave them on. A rigid alignment doesn't really describe everything in the image. And these kind of non-rigidities come from two sources. One is actual real motion, right? So when I'm I'm scanned at time 1, maybe my mouth is open. I'm scanned at time 2, my mouth is closed, or the angle of my head is different, or I'm blinking my eyes, or my tongue is moved. And so there's no rigid motion that explains, where my tongue was at time 2 and where my tongue is at time 1, and also aligns the brain. There are also distortions in MR, many of them relative to the direction of the main magnetic field. And so if the orientation of your head is slightly different in one time than another, you'll find these distortions are different in the 2 times. And so you will not be able to align the entire image volume. And the solution that we've come up with to this problem is to use what are called M-estimators. And these are kind of stolen from robust statistics. So if you do kind of the standard thing where I say, I want, for every voxel at time 1, I want it to match a voxel at time 2, what you'll find is that the transformation between time 1 and time 2 will be a little bit wrong everywhere. And the reason it's a little bit wrong is that it's trying to make the jaw alignment a little bit less awful and the tongue alignment a little bit less awful. And that will bias the alignment everywhere else. And so if you instead use these robust statistics, what it'll do is it'll find regions of the image that it can align, and it will align those perfectly and then not try at all to align things elsewhere. So here's an example. So this one I think is going to be, this one's correlation ratio, which is kind of a standard registration. Whoops, go back and forth. So this is from FSL, and this is our alignment. And if I go back and forth with them, you can see that the correlation ratio is not wrong. I mean, we would all probably call this an accurate alignment, but the brain is jumping around just a little bit. Right? Whereas here, you can see there's no attempt to align jaws and tongue. It's automatically found these regions and discarded them, but the brain is rock solid. Okay? And these are small differences, but it enables us to look for small changes across time. Okay, so I wanted to start with one clinical study that we found quite depressing that we did recently. This is with Martin Reuter, Elizabeth Gerster, and Adel Repolino. And this, I think, is my canonical example of what I was saying before about how we have not provided the tools to radiology that are really needed. So RANO is the response assessment in neuro-oncology. It's kind of the standard method by which clinicians assess tumor volume. So tumor volume is hard to assess, and so really what clinicians do is they assess tumor area. And the essence of the procedure is to find the slice in which the tumor is maximal, and then you draw a line through its biggest extent, and then you draw another line perpendicular to that. You multiply those two, you get an area, and this is a marker for how big the tumor is. So what we did is we took a bunch of scans. These are scans that Elizabeth had collected. These are serial scans of subjects with GBMs. And we used the registration across time to figure out where the head was in the scanner. And then we took the baseline data and we resliced it into the subsequent coordinate systems. Right? So we took one data set at baseline and we resliced it 7 times as if it was acquired in these different at these different locations in the scanner. And then we gave the data to Elizabeth and Otto Repolino, and they did this RANO assessment on it. And in the standard RANO, if the tumor volume has grown by more than 25%, which is this dotted line here, then we consider them to have progressed. The treatment has been ineffective. Okay? So here we take our baseline data. There's no new data, right? It's just baseline data. We take the baseline data, we reslice it into these subsequent coordinate systems, and then we give it to Elizabeth and Otto, and they do RANO on it. And these are the results. So these are simulated visits. So visit 1 through visit 7. This is the smallest volume. And you can see, and each one of these colored lines is a different subject. And so under this assessment criterion, every single one of these subjects would be deemed to have progressed, right? Their tumor has grown significantly by more than 25%. But this is the same data. This is baseline data. Nothing has changed here. There should be no change in the tumor volume. And it's not that Elizabeth and Otto are doing this wrong. They're doing it exactly right, but it is a terribly broken method by which we assess tumor volume. Okay? Okay, so I found this to be a shocking study. Okay, so on to constructing models of MRI. From MRI, this is kind of old stuff at this point. We can take any relatively high resolution, probably 10 years ago we called this high resolution. Today, I don't know, we would call it medium resolution. So this is something like a 1 millimeter isotropic MP RAGE. We can strip the skull, we can label the white matter, we can find the pons and the cerebellum and disconnect the hemispheres, and then we can cover them with a bunch of polygons to generate surface models, both of the gray-white boundary, the Pial surface. We can inflate it like a balloon, so the stuff that's red here, we're folded in, the stuff that's green, we're folded out. And we can also assign a neuroanatomical label to every voxel of the brain. So here's pallidum, putamen, hippocampus, amygdala, et cetera. So this stuff is pretty old at this point. It's fairly industrial strength. We've analyzed, I don't know, probably close to 100,000 images with it. We're constantly working to extend kind of the level of detail and breadth of these models. So for example, in the medial temporal lobe, we've been doing high resolution imaging to image hippocampal subfields and amygdala nuclei. We've been looking at brainstem and midbrain structures, cerebellar parcellations, and also white matter damage. So a lot of this stuff is kind of in the pipeline, and we're constantly working to extend these. The way we do inter-subject registration is not based on image intensity. So this is kind of the standard in the field, is to use Talairach averaging. Talairach means different things these days to different people. In general, it means a somewhat low-dimensional volumetric transform that is one way or another usually biased to structures in the middle of the brain and along the midline. If you average a bunch of subjects in Talairach coordinates, you'll find it looks like this. You've thrown away all the resolution that you spent your time in MR acquiring. So you can't even see the difference between cortical folds. In contrast, what we do is we use cortical geometry to drive registration. So we build models of the folding patterns. We represent them on the unit sphere. So here, red are things that were geometrically folded in. This is the Sylvian fissure. Here's the central sulcus. And the game we play is to take each one of these red-green patterns, extract statistics across subjects. So this is 4 subjects, represent them in some coordinate system, and then move these red-green patterns around so that they're in alignment. And at the end of the day, what we've done is we've developed a coordinate system that has kind of morphed itself onto the details of the individual neuroanatomy. It's normalized out the differences in folding patterns. And the reason this is important, which I'll show you in a little bit, is that folding patterns are predictive of cortical function and areal identity. And so if we align cortical folding patterns, we actually do a radically better job at aligning homologous functional areas across subjects. And I'll show you some evidence of that in a little bit. And here's just kind of a hand-wavy example to show that if we average subjects' data in this coordinate system, we retain, for example, the distinction between the posterior and anterior rags of the central sulcus, right? Because they're far away along the surface. They're far apart on our coordinate system, even though they're close to each other in space. We've been working a lot on longitudinal analysis recently. So in longitudinal analysis, we have serial scans of a subject over time. And we should be able to use the serial nature of this imaging in order to reduce noise and increase reliability. But when we increase reliability, we have to be careful because the entire point of this is to increase sensitivity to disease effects. So it's very easy to produce a completely reliable picture. You can just label everything 0 and everything will be perfectly reliable across time, but now you have no sensitivity to change. And so we've been working on these techniques that allow us, so this I subscript here indexes time. And so the game we want to play is if I have a set of images, I sub j, where j is at different times, can I estimate the image I, what it would have looked like if there was no, so I want to reconstruct an image I that is less noisy than the one that I acquired. And if there was no anatomical change, this would be easy. I could just average across time, and the average would look a lot less noisy. But of course, there is anatomical change, and that's what we're trying to find. And so we use these kernel density estimators to allow us to get noise reduction in regions where we expect no change, but preserve anatomical change where we see it happening. So I'll show you an example of this. This is data from the ADNI study. So this is a single subject who converted at month 0. So you'll see that the time goes from negative to positive. At 0 was conversion. And so in this image, over time, you can see that things are happening, right? The ventricles are getting bigger. There's a cortical atrophy. The hippocampus is getting smaller. Ventricles are getting bigger. But lots of other stuff is wiggling around as well. And it's this other stuff that makes it difficult to detect on, say, a 6-month time frame what's actually happening. Because these changes here, where things are jumping up and down, are probably differential B0 distortions across time. And so which change is real and which is not? If I give you this full ensemble across 2 years or something, then we can see the pattern emerge. But our goal is to find it over 6 months or 3 months. And so if we use these kernel density estimators, you can see it really removes a lot of the uninteresting variance while still retaining, you can see the ventricular volume increasing and the hippocampus shrinking here and cerebellum shrinking. Okay. We've quantified the effects of this. So this is retest reliability. So this is percent difference in a structure volume in scan-rescan. The percent difference should be 0 in this case. There's no anatomical change in these short time frames. And so the green is cross-sectional and the yellow is longitudinal. And so you can see we're really down here under 2% for most structures, in the 2% range. And so we're somewhat confident that across time, if we take care on the imaging side, we can detect changes on the order of just a couple of percent. We applied this to the full ADNI study. So here's 4 groups of subjects. These are elderly controls, people who were MCI at baseline and remained MCI throughout the study, MCI who converted to Alzheimer's, and Alzheimer's. Each different line is a subject. This is age on the horizontal axis and hippocampal volume on the vertical axis. And so you can see that there is this slight decline across time in the controls. The MCIs are going down at a steeper pace, and then the converters in Alzheimer's subjects really look the same, this kind of sharp decline in hippocampal volume. And the nice thing about this is that it really can power clinical studies. So if you look at just, so this is the percent of subjects that we would need in a longitudinal analysis relative to a cross-sectional one. So for example, if thalamic volume was our biomarker, and we needed 100 subjects in cross-sectional to power a study, we would only need 20 using a longitudinal analysis. And it really does enable you, in brain imaging, we typically think of cross-subject variance as the largest source of variance. And in a longitudinal study, this largest source of variance drops out, right? Because we're looking at change instead of absolute measures. And so longitudinal analyses really can dramatically increase your power to see effects. Anything on that? No questions? None? Okay. So more recently, we've gotten a PET scanner at MGH, and we've been working a lot with also, with Gitte Knudsen, who's in Copenhagen. And one of the nice things about the cortical models is that they enable you to kind of do guided smoothing. And I'll tell you what I mean by that in a moment. But if we take just a binding potential map, so these are kind of serotonin receptor density maps in the volume, and the typical thing one would do with such a thing is to smooth it, right? And if we smooth it in the volume, as is typically done, and we sample it onto a cortical surface, what you can see is the cortical folding patterns. And this is a bad thing. This means that my volume smoothing has biased the estimates of the binding potential. So for example, here's the central sulcus, here's the periotemporal sulcus, here's the periotemporal gyrus. And this is because when I do an isotropic spatial smoothing kernel, I'm differentially smoothing from different classes. Right? So I'm smoothing from the gray matter, which is what I want to do, but I'm also smoothing from white matter, or from CSF, or bone, or the Hudson River, depending on how big my smoothing kernel is. And because cortical geometry is not uniform, this isotropic spatial smoothing induces biases in the estimation. Right? So the binding potential down here in the insula should not be this high. This is really the basal ganglia spilling out into cortex across the extreme capsule because it's only a couple of millimeters away. And so one of the things we can do with the cortical models is we can essentially remove this effect. So we can smooth along the cortical surface where we're guaranteed, no matter the size of our kernel, to always be smoothing within cortical gray matter. And so we remove this problem of smoothing with stuff that's essentially zero and corrupting our data. Okay? And this actually survives. The effect that I was showing you survives even in group maps. So this is 16 subject group map. One of the fun things happening in MR these days is people have really been pushing the resolution. The physics in general will tell you that you lose signal as the cube of your voxel size. So if you have your voxels, then your SNR goes down by a factor of 8. And that's true for certain types of noise, but it doesn't really apply to the dominant sources of noise in fMRI. And so people have had a lot of success making their voxels significantly smaller without paying as steep a price as they thought. And so with voxels going down to a millimeter and below, we're starting to be able to probe not just where am I across the brain, but where am I within the cortical ribbon? Am I in deep cortex? Am I in superficial cortex? And this is joint work with John Palomini and Larry Wald. And so we've been working to extend the cortical surface models in order to allow this type of laminar analysis. And at the moment, what this amounts to is that we're not just modeling the gray-white boundary shown here in yellow and the Pial surface in red, but we evolve a family of surfaces between them that allow us to sample at different cortical depths. Okay? And so this actually grew out of John's PhD work. He did a PhD at Boston University with Eric Schwartz. So one of the things that we've known about the human visual system for 1977, 40 years or so, is that there's a pretty good closed form approximation to the mapping that goes on between the retina and primary visual cortex. This is kind of an astonishing and somewhat little known fact in neuroscience, that there's this a closed form solution that tells you if I treat the retina as a complex plane, x plus I, y, and I treat visual cortex as a complex plane, u plus iv, then the mapping that tells me where a coordinate in the retina goes onto cortex is well approximated by a complex log map. And so John had done a PhD on this, actually with me and with Eric. The complex log map induces distortions, geometric distortions. It's also not translation invariant. And so if this is the retinal plane and this is the cortical plane, then this complex log transformation, if this is the fovea, would take this stimulus and make it look like that. And so John's goal, and it's important to have goals, was to paint the letter M on the surface of primary visual cortex in the human brain. And so what he did is he took this complex log transform and he measured the parameters of it, and he essentially inverted it. And he said, what stimulus do I have to show to the fovea in order to give rise to this pattern of activation in primary visual cortex? Okay? And so if you do that and you do your kind of standard subtraction maps and paint it onto a folded cortical surface model, you get something that looks not much like anything. And the reason is that the folding patterns, kind of this extrinsic coordinate system imposed on the cortex, obscures the simple functional response of the brain. And so if we take this folded model and we inflate it like a balloon, here's the letter M, clear as day. And so this is really the motivation for why we want to generate cortical models, and why if you're doing functional analysis or molecular analysis of cortical properties, you really want to do it with a cortical coordinate system, with the anatomical models. Even if the anatomy is utterly uninteresting to you, which it is to most people, having access to the models of the anatomy enables you to do this type of analysis. Okay? So this very simple pattern is lost in kind of the arbitrary slices that I typically acquire my data in and visualize it in. And of course, once we painted an M on the brain, we had to paint a G and an H. Okay, and so there is a little bit of, okay, it's pretty cool to just do this, and I'll admit that was some of our motivation in doing it. But there's actually some science to be done as well. And so what John did is he took these, so we chose M because it's an asymmetric stimulus, and we're able to see kind of departures in the veridical nature of the representation. And so he took this fMRI pattern and he sampled it at different cortical depths. So this is near the gray-white boundary, this is near the Pial surface, and these are kind of the middle layers. And so the color reflects kind of the SNR, how much signal do I have? And if you look at these response patterns and all you were doing was assessing how much signal do I have, which is pretty much the standard way we assess fMRI data, right? We look at SNR or temporal SNR or something. Then you would say, oh my goodness, look out here. This is where I want to be sampling my signal. I have giant amounts of power. But actually, out here, you can see that the fMRI signal is not itself a veridical representation of what the neurons are doing. That is, the M has been corrupted. Probably there's a Pial vessel running here and maybe another one down here. And so much of the M has been lost if I sample out at the Pial surface. And you're probably better off going back a couple of millimeters, actually probably about 3/4 of a millimeter, and sampling somewhere in the middle of the cortex, where I still have power to see effects, but it's more accurately representing what the underlying neurons are doing. Okay? Okay, on to connectional analysis. So I wasn't sure how much people know about diffusion MRI, if people in general know what's going on with diffusion. So this is actually a GIF that Larry Wald gave me. This is from our Connectome scanner, which is a 300 millitesla per meter scanner we have in Charlestown. It's an astonishing technical feat that Larry and Siemens accomplished in order to get this thing working. And what the GIF is showing you are these orientation maps. So in diffusion imaging, we don't acquire scalar images. We acquire orientations at each point. And we can acquire multiple orientations in order to try and get an idea of where axons are going in space. Okay, kind of a simple idea. And the game of diffusion tractography is to take these different orientation maps and draw lines through them, right? And to kind of stitch them together so that I get models of major fiber tracts in the brain. And the problem with diffusion tractography is that it's a terribly ill-posed problem. That I can take a single wrong step due to noise, and now, I'm on 95, and I think I'm headed to the GW Bridge, but instead I've gone on 95 North. I missed the Cross Bronx, and I wind up in New Haven, right? So a single wrong step, you can have beautiful data, and you take a... No, if I'm crossing the Throg's Neck, and I take 95 North instead of the Cross Bronx. Anyway. Right, so a single wrong step can doom you. And can doom you in massive ways. You can have great data with a single point of noise, but the stuff that you get out of it is completely and utterly wrong. And so we've been trying to use the anatomy in order to guide the creation of these anatomical structures. This is work from Anastasia Yendiki. So she's called a tracula. Tracts constrained by the underlying anatomy. And the basic insight here is that the tracts don't occur in a vacuum. They occur, so if we take cortical spinal tract as an example, cortical spinal tract runs from primary motor cortex through the internal capsule, lateral to the thalamus, medial to the pallidum, into the brainstem. And we know where all those structures are in the brain. And if we know where all those structures are in the brain, there are very few degrees of freedom left for the spinal tract, for the CST, right? It can't go across the ventricles. It doesn't go, down into lateral temporal cortex. And so what we do is we have a training set that a neuroanatomist, and in this case Gina Gustanak, sits down and visually validates, so models of the cortical spinal tract, and then we extract probabilistic models that relate the fascicles to the surrounding anatomy. So what structures do I pass near? Am I lateral to them? Am I medial to them? This kind of thing. And this enables us to build a procedure that is completely automated. And so we can generate these models of, what is it now? Let's go on this slide. Something on the order of 17 or 18 fiber tracks automatically. So the game you play is you have your T1 weighted MR, you give it to us, we generate models of the anatomy, then you have a diffusion scan, and you give us the models of the anatomy and your diffusion scan, and we go off and build models of major fascicles. And the nice thing about this procedure is because all of the statistics that it's based on are relative, that is, we're not saying where in space is something or how big is it, we're just saying what's nearby it. It's pretty invariant to atrophy. And so this was work we did with Diana Rosas, where we looked at Huntington's disease. So this is 55 subjects or so. It's completely automated, and we're able to extract disease effects. And this is the case whether we include Huntington subjects in the atlas or not. So if we build an atlas off of healthy controls, we can still generate accurate models of, say, the cortical spinal tract and Huntington's and find changes in, let's see, what is this, radial diffusivity or axial diffusivity or fractional anisotropy in these different fiber tracts. We can play the same longitudinal game with these tools. So in diffusion, because diffusion is so ill-posed and so sensitive to noise, we actually get a bigger win here. So we get about a factor of 10 decrease in the number of subjects that we need in order to see effects in serial diffusion scans versus treating them cross-sectionally. So this is, again, 43 Huntington subjects. This is the forceps minor, so left hemisphere, right hemisphere. Here it's going through the callosum. This is if we treat it as a cross-sectional study. So every one of these little points is across time. So this is position in the tract as I go this way, and then every little line representing what's going on across time. So this is fractional anisotropy, and you can see at this point in the forceps minor, the FA is going up a little bit in the cross-sectional analysis. But if I do it longitudinally, you can see that the slopes are really almost consistently downwards, and I have significance across the entire extent of the forceps minor. Okay. Okay, on to architectonics. So do people in general know what architectonics are? Okay, so architectonics have a history of hundreds of years in neuroanatomy. The most famous is certainly Corbinian and Brodmann in 1909. And essentially what the game that is played inside architecture is that we take the cortex and we divvy it up based on microscopic properties of the tissue. So what's the density of cells? What's the distribution of cells in different layers? Do I see big cells in this layer? Do I see small cells? Is layer 4 present? Is it kind of there? Is it completely absent? And so Brodmann took the brain and he divided it up into about 45 different areas based on these properties. And so you can see, here's Broca's area, here's area 44, and here's area 45. Here's primary visual cortex, area 17. And the properties that define these areas are too small to see in vivo. They're, in general, kind of the density and laminar distribution of mostly of cell bodies, but also of myelin. And so this really was seminal work that drives a lot of the field. It's the way many of us think about cortical areal identity. We call it a Brodmann area. We'll say, we're in area 45 or something. But Brodmann did a single brain, right? Rather, he published a single brain. We don't even have pictures of the brain. We have colorized drawings. So here's 44 and 45. We have no idea of variability, right? Is 44 like this most of the time, but sometimes it's like that? Probably not. But we actually don't know. And so in neuroimaging in general, I would argue that there are two primary techniques for assessing areal identity from an image. And so there are some tools around that you can give it a Talairach coordinate and it will tell you, well, I think it's 44, but it could be 45, it might be 6, it might be 9. But by far, the more common method is this one. And I don't actually mean to make fun of this because anyone who's grown up around neuroscience will do this, right? You cannot help it. You say, well, it's a language task, it's in the frontal operculum, I'm pretty sure that's Broca's area, I'll call it 44. Right? But these assertions come with no ability to assess uncertainty. We have no idea, is it 90% 44 and 10% 45? Might it be 6 and 9? And so recently, there have been a series of studies, mostly out of Germany, from Karl Zillis and Katrina Muntz and their group, in trying to resolve this problem. And what they did is they engaged in the incredibly labor-intensive task of doing serial histology across the entire human brain and somewhat kind of semi-automatically defining these architectonic areas and looking at their variability. And these are images I stole from Katrin from a set of different studies. So let me take a step back. And so what they did is they took their brains. They had an army of graduate students and postdocs manually or somewhat automatically label cortical areas. So they would label, for example, area 17, primary visual cortex, in a set of subjects, 10 subjects. And then they take their 10 subjects and they align them to some coordinate system. Okay? And then at every point in the brain, they just count and say, how often was this area 17? And if the coordinate system is perfect, right, if the coordinate system is exactly correct, then what it will do is at every point in space, the value you'll get is either 0 or 10. So you're perfectly certain you're out of the area, or you're perfectly certain you're in the area. This would be a perfect coordinate system for aligning architectonic areas. Okay? But what they found in a series of papers in Brodmann Area 17 and 18 and 44 and 45 is that this Talairach type of registration is terrible at aligning areal identities. If we look at Brodmann Area 17, which is by far the most stable architectonic feature of the brain, it's always in the calcarein sulcus. You can see the stria of Gennari by eye. It is the most prominent myelo-architectonic feature of the brain. Even for Area 17, there's this tiny little dot of red in the middle, right there. Sorry, I've had a fair amount of coffee. Where all 10 subjects align, but the majority of the distribution is out here around 5 or 6 subjects. So even for Area 17, there are almost no regions where I have more than 5 or 6 subject overlap. And it completely falls apart if I just go one step up the hierarchy. To V2, to Brodmann Area 18, you can see that the majority of the distributions are down here, like 3 or 4 subjects. And if I go out to language areas, so 44 and 45, you can see that probably about half of the distribution is blue. Right? So that means that no 2 subjects agreed. And I actually think that this is a major reason, actually Nancy Kanwisher, who's at MIT, has been looking into this, that this is one of the major reasons why we've had problems studying language in human beings. It's because our coordinate systems are quite poor. And so it's very easy to do a language study where you have 30 subjects, and every one of them gives you a robust activation, and your group maps show nothing. And this reflects the fact that the coordinate system we're using to analyze the group data is a terrible coordinate system for analyzing cortical effects. Okay? So Katrin was kind enough to give us access to this data. We did essentially the same study, but instead of aligning to Talairach coordinates, we painted onto surfaces, and we aligned folding patterns. And if you look at the results, so sorry, I've changed color scales here now, so this means no subjects overlapped. This means all 10 overlapped. If you do this study and normalize out cortical anatomy by folding patterns, what you find is that the folding patterns are perfect predictors of some architectonic areas. It's not that the architectonics are just mysterious and unknowable. It's that the coordinate system we were using was the wrong one. And that so for primary visual cortex, you can see that the alignment is pretty close to an ideal one. There's only about 2 millimeters of uncertainty here at the edge. But other than that, we're perfectly certain we're in the area and perfectly certain we're out of the area. And 18 is not too bad. It's pretty much the same. And even if I get out here into language areas, 44 and 45, you can see that, yes, there's a lot of spread here in the distribution, but there's this large core of the area where I'm perfectly certain that I'm in area 44 or 45. Okay? Yeah? Yes.
What is the medial surface?
We don't have a lot of, so I mean, here's some medial.
All right, in front of you.
So we don't have any areas from them in the frontal medial surface. One of the things that I'm pretty convinced of is that a lot of this spread here is errors in the manual labeling of the areas. The distinctions between, say, 44 and 45 are very subtle. And it's very hard to see the histological features that define these boundaries. And I'll get back to that in a second. Okay, so this stuff was done with data from Carl and Katrin. It was unbelievably labor intensive. They did it histologically, so they physically cut the tissue. When you physically cut tissue, you introduce distortions that you can never remove again. You've torn it, you've mounted it on a stain, it shrinks. You've mounted it on a slide, it shrinks, you stain it. And so you have two high resolution dimensions, the in-plane dimensions, and then a through-plane dimension that are very difficult to do anything in. So it's intrinsically 2D data, which causes difficulty in the analysis. So we were very interested in doing this with MR. I mentioned before that there's this relationship in MR between SNR and voxel size. So if we have our voxels, the SNR goes down by a factor of 8. To recover that factor of 8, I have to scan 64 times as long. And so it's not feasible just by length of scanning to get to the resolutions that we need on the order of 100 microns to start to see cortical architecture. So instead, what we've been doing is scanning ex vivo. When we scan ex vivo, we get about a factor of 1,000 in SNR. We have special built small coils. This is in collaboration with Larry Wald, that we put right on the sample. There's no skull in the way. There are no cardiac and respiratory cycles messing up the data. We can scan for 40 hours, which we do routinely. And we can take that factor of 1,000 in SNR and trade it off for about a factor of 10 in resolution. So this is 150 micron isotropic imaging through the primary visual cortex. Here's the stria of Gennari in layer 4, this myelinated stripe. The stria of Gennari is, as I said before, by far the most prominent architectonic feature of the brain. If you want to do architectonic imaging and you cannot find the stria of Gennari, then you should do something else with your life. But with about 15 minutes of imaging, we can see it quite easily, and we can delineate the borders of primary visual cortex. I've been working with Brad Hyman and Gina Gustenak, looking at other cortical areas. This stuff is actually reasonably old at this point as well. So we went looking in entorhinal cortex. This is a photomicrograph of a Nissl stain. So this is the gray-white boundary. This is the Pial surface. Entorhinal has this very interesting cortical architecture where the layer 2 neurons come in clumps. So you can see this kind of cell-dense clump in a cell-poor region, a cell-dense clump. These layer 2 islands are also one of the earliest locations of Alzheimer's pathology. So they're devastated in the clinically mildest form of AD, post ex vivo diagnosis, postmortem diagnosis. So you can see the neurons are devastated here. And so we went looking for these layer 2 neurons in ex vivo MR. So this is 100 micron isotropic ex vivo MR of the medial temporal lobe. Here you can see the layer 2 islands in entorhinal cortex, these little cell-bright blobs. You can see the molecular layer in the hippocampus. You can see all this laminar structure in the hippocampus. And you can do things that are very difficult to do histologically because this data is isotropic. I can stick a plane in layer 2 and look at a tangential section and see each one of these entorhinal islands. And then we could do the same game that we did with the histological data. We could build population models and use them to assess the degree to which folding patterns predict these areal boundaries. And so can the stuff that I see, or rather, is the stuff that I can see, folding patterns, predictive of the stuff that I can't see, these architectonic boundaries that are critical to find if we want to, for example, diagnose Alzheimer's a little bit earlier? And the answer is yes, that there is some spread in these distributions, but there's a big core of the area where I'm absolutely certain I'm in entorhinal cortex. And so we can do the same thing we did before. We can take these and we can map these to in vivo subjects. So this is a study we did with Randy Buckner. So these are the clinically mildest in vivo Alzheimer's subjects. They're a clinical dementia rating of 0.5, so they're very mild. And so we took these subjects, we predicted where entorhinal was, and then we looked at cortical thickness in patients versus controls. And so this is left hemisphere and right hemisphere. This is cortical thickness. These are Alzheimer's subjects. These are controls. And if I look at entorhinal cortex, where we predict these boundaries to be, you can see that there's already this dramatic thinning between the Alzheimer's subjects and the controls. And if I take my ROI and I translate it a centimeter posterior, this effect largely goes away. So it's not the case that all of temporal lobe is thinning, and it doesn't matter where I stick my ROI. In fact, we have some confidence that we're finding the boundaries of this invisible area where we expect these very early disease effects to be. Okay? How are we doing on time? Okay, still okay. So I wanted to describe some stuff that's been going on here. So this is a U01 funded by the NIH and the NFL. Wayne Gordon is the PI here, and Kristen Dams-O'Connor is involved in this. Wayne and Kristen and I have been on about 1,000 TCons together, but we have never met until today. So I'm excited to have lunch with them. And this was a great opportunity for us to try and push for some interesting results. And the basic idea of the study is to try and assess the effects of traumatic brain injury in vivo. And we're kind of using ex vivo MRI as a link. So we have a bunch of studies where we have in vivo scans of people who have had TBIs, they've lost consciousness. And we're going to do histopathology with a group of really pretty amazing neuropathologists that have access to whole brain histopathology. And ex vivo MR is going to act as the link between these two. That is, we're going to take these people who've come to autopsy, we'll scan them ex vivo, we'll use them both to find effects in vivo that we might not have found because we have such better data ex vivo, but also to guide where we do histology, because we're not going to look under a microscope at every single square millimeter of the brain. We have one big problem to solve from our perspective, which is that the process of so death and brain extraction and fixation induces pretty large-scale geometric distortions in the brain. And so this is a scan of a subject in the study. This is the in vivo scan, and this is kind of the ex vivo scan. I'm happy to explain to anyone what this actually is, but it is essentially an ex vivo MR of the same person. And you can see that there's this dramatic change in the geometry of the brain due to kind of the physical process of both autolytic processes, but also the physical extraction of the brain. And so we need to correct for these distortions if we want to make pixel-wise use of the ex vivo scans, in order to find what's going on in vivo, and then furthermore to guide the neural path. And so one nice thing for us is that the topology of the folding patterns is preserved by death. And so here's the in vivo scan, here's the ex vivo scan. Again, you can see this large geometric distortion. But if you pick out any of the folds, so here's the central sulcus, you can see that the shape of the central sulcus is the same. It's maybe changed its angle, but all the secondary and tertiary folds and the shape of the sulcus in general has been preserved. And what that means is that our cross-subject registration procedure that's based on cortical folding patterns, this is actually an easier problem than cross-subject. Right? So registering this to this is easy compared to registering it to a different set of folding patterns. And in fact, it works pretty much out of the box. So we can take our cortical registration, establish point correspondence between this subject, this surface, and then build a biomechanical model of what kind of the rest of the brain, the interior of the brain, would be doing, and use it to drive a nonlinear registration that gives us accurate point-to-point registration between the in vivo scan and the ex vivo scan. And it's pretty cool. So here's an example. This is the in vivo scan warped to be in register with the ex vivo, and here's 200 micron isotropic ex vivo image that we got from our 7 Tesla scanner. Right? So the registration's pretty good. And furthermore, we can go and give the data to neurologists like Brian Edlow, and he can find regions in the ex vivo where there's, for example, lesion white matter, and this enables us to go back to the in vivo scan and say, can we find anything there? And in fact, we can. So if we go back to the in vivo scan, we can see this kind of subtle darkening that indicates a lesion in the white matter that we would never have noticed in vivo. So I'll show you one more example. Here's another lesion in the white matter that we see clear as day in the in vivo. And then we go back into the ex vivo, and we can see, yes, there's some darkening in the white matter indicative of the lesion. Okay, so that's all for ex vivo MR. So I sat down one day, I don't know, maybe a year ago, and I decided to write a list of the things that we don't know about the human brain. And this is not meant to be an exhaustive list, because it's kind of astonishing, the stuff that we don't know. Even after decades of amazing imaging, there are fundamental things about the brain that we don't know, right? So I showed you the Brodmann area maps. How many Brodmann areas are there? Are there 40? Are there 60? Are there 80? Are there 100? We don't know. Are the Brodmann areas hierarchical? You know, is 44 and 45 higher in some, graph theoretic sense than 17? I guess so. We think so, but we don't really know. Are the higher ones more variable? There's some evidence that this is the case, but it's very sketchy. Maybe. What's the relationship between connectivity and Brodmann areas? We have theories about this, but we actually have no idea. Here's my favorite one. Why do fibers run together in fascicles? Why do axons run together? Are they lonely? Right? There must be some physiological benefit for axons to run together in fiber bundles. There are axons, Suzanne Haber has this very cool data where she shows axons that will enter into the uncinate fasciculus, and they'll run with it for a while, and then they'll head off into parietal cortex. They go centimeters out of their way to hang with their friends in the uncinate. Why? We don't know. I mean, it's a good thing they do. Diffusion MRI would utterly fail if there weren't for fascicles. But we have no understanding of why fibers run together in fascicles. How many neurons are there in the brain? 15 years ago, we would say 100 billion. Somehow that's been downgraded to like 84 billion. We lost 16 billion neurons. Right? We have no way to count them. The techniques that assess things like number of neurons are extremely indirect. Right? We kind of stir it up in a blender and weigh it and measure different components of it. We don't actually have a method that allows us to count individual neurons. Okay? So I've been working a lot recently with David Boaz, here's David, and a group at MGH who does optical imaging in order to do kind of 2.5 dimensional imaging, and I'll tell you what that means in a minute. And so, so as I said before, high resolution MR comes at this terrible price. We have played a lot of the games that we know how to play to get these orders of magnitude increase in resolution, and I don't think that there are games left to be played. Okay, we can get to 80 microns maybe, but we're not going to get to 10 micron over large sections of, over entire cortical hemispheres anytime soon. The SNR is just prohibitively low. And so we've been working with this technique called optical coherence tomography. OCT is an interferometric technique. It yields micron scale images of the tissue. And what's critical about OCT, and the reason it's like $1 billion image, industry in ophthalmology, is that the laser light in OCT penetrates tissue a couple hundred microns or a millimeter. And what that means is that we can image tissue before we cut it. right? We can use our OCT rig in order to image under the surface of the tissue and then remove the stuff we've already imaged. And you might think, okay, image then cut, cut then image, what's the big deal? But it's everything. It means that we're imaging undistorted tissue, and we can generate 3D undistorted representations of the underlying tissue. Once you've cut the tissue, you have introduced distortions that you will never correct, accurately enough to enable you to, say, follow a single axon. Right? It's just, in my opinion, it's just not possible. And so the OCT data has been stunning. This is 0.9 micron in-plane OCT of the human medulla. It looks just like the atlas. And the ex vivo MR kind of made the in vivo MR look like a cartoon, while the OCT makes the ex vivo MR look like a cartoon. So if I zoom in on the OCT, you can see that it shows both fiber bundles, and these little bright dots are neurons, which I'll show you in a minute. We've done a bunch of preliminary studies in the OCT. We've shown that, that we can essentially extract the same information as a Nissl. So here's a Nissl-stained image. This is an OCT. We can see all 6 cortical layers. We've kind of verified this with an anatomist, with Gita Gustanak, who sat down and labeled the layers, both in the OCT and in the Nissl. And they're essentially in the same place, modulo distortions in the Nissl. And then I just wanted to give you an idea of how stunningly high resolution the OCT is. That it's not just high resolution, it's undistorted and large field of view. And so here's 3 different ROIs, a little bit of the internal capsule. Here you can see the fiber bundles as they bend, coming into the cortex. And then these little bright dots are neurons in the gray matter here. So here's an image of medial temporal lobe. There's a low resolution image in the background. This is a 2.9 micron image here. And then there are 2 high resolution insets that I'm going to zoom in on. These are 0.9 micron in plane. And I'll just zoom in all the way. And so we can see these, mossy fibers in the hippocampus, which is pretty cool. There's another game that one can play with OCT. We can take advantage of birefringence. So birefringence is the optical property of a material having a refractive index that depends on the polarization and propagation direction of light. So we can stick polarized light into the tissue, and then the polarization of the light that comes back tells us something about the orientation of the tissue. So essentially, we can get something that's like a diffusion tensor scan with the OCT. This is work that Hui did with Tanner at UMin before she came to MGH. Right? And so myelinated nerve fibers are birefringent. And we can use the retardance, so the phase change in the polarization of circularly polarized light to map out fiber architecture. So here's an example. This is 15 micron isotropic OCT in the human medulla. This is the retardance, which is kind of loosely like an NFA map. And this is 300 micron isotropic diffusion scans on our 4.7 scanner of the same tissue. I'll just fly through this. And so what you can see is that, again, even though we're cutting here, we're cutting after we image. And so the cutting has introduces no distortion into the imaging. And we can, I'll run through one more. And you get these kind of perfect alignment slice to slice as I go through the tissue. And then we can do the same thing with orientation. We can map out primary fiber orientations. Again, this is 15 micron isotropic. This is 300. So this is, what is that, a factor of a couple thousand, I guess, in voxel volume. We've looked at the high resolution OCT. So this is 0.9. This is using a 40x objective here and compared it to Nissl stains. And we wrote a little segmentation procedure to enable us to pick out neurons in the Nissl and neurons in the OCT. And then we just overlay them. So the green, which one's which? The green is the Nissl, the red is the OCT. And so you can see that there's quite good alignment of the Nissl and the OCT, and that we are in fact seeing neurons with the OCT. And that any residual mismatch here is almost certainly residual distortion in the Nissl through the process of cutting and mounting. And then finally, we've been looking at building automated procedures, because now we're able to acquire volumes of tissue that are undistorted, that show individual neurons. And so this is Ender Kunikulu's work. This is entorhinal cortex, I should say. So this is layer 2. These are the layer 2 islands. And so we're building tools that enable us to do actual counting of neurons over large, extensive tissue. And then if we zoom on the OCT even further, in entorhinal cortex, it's known that there are 2 classes of large neurons. There are inhibitory stellate neurons and pyramidal neurons. And if we zoom in on the OCT, you can really clearly make out the shape of these neurons, right? So here's a pyramidal neuron, this kind of classic triangular shape. And here's the stellate, the 5-sided stellate. Okay, so that's all. I tried to mention people, lots of people contributed to this as I went through. And I'm happy to take questions. Any questions on that whirlwind tour of the brain?
About your data on retrospective correction based on the bad scoring. So when
Sorry, which one? The best based on
the first part where you are reacquiring the part
sure.
of case
At the very
space that
beginning. That was so long ago, I forgot about it. Yeah, okay.
that had bad scoring. So essentially, that will reflect the breathing pattern of your patient. Is that correct?
No,
Or
it's more likely small readjustments. I mean, typically, what you see, at least in reasonable subjects, is not. So we cannot correct for ballistic movements of the head constantly, but typically what you'll see is someone will have an itch and they'll scratch or they'll readjust their head a little bit. So it's mostly small readjustments of the head in the coil. So it's not cardiac. So cardiac and respiratory are different things and can be measured differently. There are navigators for that. There are bellows. But that's not what I'm showing here. This is actual motion of the head, not distortions of the magnetic field.
Okay, so regardless for what it is, have you analyzed over time with multiple patients to see whether there is a pattern that could drive you to actually change the strategy in how you are acquiring case space, so you are reducing that effect?
No, I don't think that we've done that. I mean, it's fairly early days. We've installed the sequence both at the main hospital and at Children's Hospital, where it's in routine use now. So there are motion logs so the sequence also outputs motion logs that it detected. But I don't think anyone has done that. So so we would try and
I'm just thinking.
Collect the center of case space first or last? Or?
Well, I mean, the central space is going to influence your motion a lot greater than the high frequencies. So if you sort of have a pattern there, then you could sort of sparsify the way you are encoding so that it reduces it.
Yeah, that's an interesting idea. We haven't done anything like that, no.
Okay. Thank you.
Thank you very much for a great talk. I was just wondering if you could comment a little bit more about the translational potential of neuroimaging. So what you described was, I think, a phenomenal amount of work in increasing precision, in mapping structure and function and producing sort of meaningful interpretations and models of the brain. But when we come to disease prediction in the clinical space, especially for diseases that do not have significant major structural disruptions like schizophrenia, Alzheimer's disease, how do you think disease modeling and disease prediction can happen? Because we cannot rely in case-controlled designs for that sort of thing.
Right. So I think, okay, so I should have a disclaimer, which is that I did start a company. So for many years, it drove me up a wall that I was in the Department of Radiology at MGH, and yet radiology at MGH had not used any of the things that we had done. Right? We had zero impact on clinical care. And in the intervening years, just kind of by being there a long time, I'm kind of friends with a fair number of radiologists, and there have been a number of cases where they'll scan a subject, they'll say, well, we think there's a dysplasia. Can you analyze this data set? And we've done that over the years. And there have been some successes. We've detected like lack of change in a brain tumor that avoided a surgery one time. We've found some dysplasias, but none of it was ever self-sustaining. And the reason it's self-sustaining is that we're not allowed to charge for it. The reason we're not allowed to charge for it is that we're not FDA approved. And the reason we're not FDA approved is that we're not set up to get FDA approval. And so this was kind of the chain of events that led us to found a company, which is really just to get FDA approval. That's kind of the goal. So in diseases where there's massive variability, like Alzheimer's, across subjects, I think that longitudinally is your best bet. I think that looking for change in the brain over short intervals is feasible. I think that we can probably detect change in an Alzheimer's brain if we're careful on the imaging side. On the order of 5-6 months, I think we can probably find things that are of diagnostic significance. I don't think that we're from the anatomy we can say yes or no. I think it's simply another fact to add to your clinical score. I think that there are some diseases like dysplasias that we can detect like neon signs. Right?
Just a question.
Yeah.
How many sites are implementing the reacquisition technique that you know?
So it's part of a lot of the HCPs, the human connectome projects. So a bunch of the HCPs have been using it. At the moment, it's routinely used clinically at Mass General and at Children's. And as far as I know, those are the only two. I think there was some discussion with some people at Cleveland Clinic, but that's all I think right now. But we'll give it away to anyone who wants it. I think you need a Siemens research license with Siemens.
WIP or a CDP or something?
I think it's a WIP. I don't think it's a CDP, but you should ask, Andre. Anybody else? Okay. Thank you. Oh, you got a present too?
Nice for you.
Oh, thank you very much.
Thank you for coming and giving a wonderful talk.
Thank you.